The Institute Gulbenkian Ciência is a private institute devoted to biological and biomedical research and to graduate training. It was founded by the Carlos Gulbenkian Foundation. IGC has small independent research groups working in an environment designed to encourage interactions and exploit synergies. It runs internationally recognized PhD programs explicitly directed towards intellectual breadth, creativity, theory, and independent scientific thought. One of these research groups, led by Miguel Soares, discovered a new mechanism of protection against malaria.
There is a glycosyl transferase that was mutated very early on in human evolution. And that glycosyl transferase is called alpha-1,3 galactosyl transferase. It generates a glycan called alpha-gal. And those mutations made that humans do not express the alpha-galactosyl transferase and as such do not express alpha-gal.
We have high level of circulating anti-alpha-gal antibodies. Here in the coming issue of Cell, we test whether these naturally occurring anti-alpha-gal antibodies confer protection against Plasmodium infection, which is the causative agent of malaria.
IGC has established a collaboration with the Institute of Tropical Medicine and Hygiene in Lisbon.
The Institute of Tropical Medicine and Hygiene from Lisbon does research on several tropical diseases and is very well known in these entomological studies, and that's why our expertise came to the project. We rear mosquitoes and infect them with Plasmodium, and this is essential, and it was essential for the results obtained.
So why malaria? Because we know that malaria played a major role in human evolution. It exerted very strong selective pressure that has selected and fixed specific alleles that we now find in human populations. It's often lethal. It's transmitted to humans via the bite of a mosquito that drops sporozoites in the skin where they stay for no more than 1 hour, let's call it.
It was already established that specific components of the gut flora express alpha-gal. Therefore, we ask whether those specific components of the gut flora induce the production of anti-alpha-gal antibodies. So we test this hypothesis in germ-free mice. So we colonize the mice using E. coli strain that does express alpha-gal or E. coli strain that does not express alpha-gal. Initially, we asked whether Plasmodium sporozoites inoculated by the Anopheles mosquito into the dermis express alpha-gal. Upon the colonization with E. coli strain that express alpha-gal, we saw that the mice produce high level of IgM anti-alpha-gal antibodies. The conclusion of this part of the study is specific component of the gut microbiota can confer protection against malaria transmission.
We know that children under the age of 3 are the most susceptible to develop malaria. What we found is that children under the age of 3 are also those that have the lowest levels of antibodies against alpha-gal. What Bati did was to immunize these mice with a synthetic alpha-gal. That is no longer relying on the presence of the bacteria, but is now using a vaccination-like protocol. He tested many different adjuvants to trigger a potent antibody response that contains both IgM and IgG antibodies against alpha-gal. And he found that using this approach, he can really enhance the protective effect of alpha-gal antibodies against malaria transmission by mosquitoes.
So Miguel, I think there is one thing that's important to be mentioned. So these antibodies are protective in the skin stage.
How do we know that?
So I did some experiment by introducing the parasite into the dermis, and then I see the protection. But when I did this experiment by inoculating this parasite into the bloodstream, they're not anymore protective against Plasmodium infection.
So that would suggest really that sporozoites must stay in the dermis for a given period of time so that some effector mechanism can go there and probably destroy them.
So we know that the anti-alpha-gal antibodies are strictly dependent complement. So for that reason, we use the complement depleted mice, and we test whether these anti-alpha-gal antibodies are protective against Plasmodium infection.
So that means that when sporozoites are in the dermis, they express the glycan, so the anti-alpha-gal antibodies bind to the surface of the sporozoites, they activate complement, and then what?
And they kill the parasite into the dermis.
So that's the whole point, isn't it? It's
Yes.
complement-mediated killing.
This institute, it has a lot of privilege, like having a germ-free facility. Without this, I think we can never finish this story as we wanted. And besides that, there's a couple of facilities like the flow cytometry and the imaging facility that are really the core of my study. So having all of these in the same environment, it makes my PhD and this study easier.